So let's talk about blood. So here's what blood looks like under a microscope. So this is a photomicrograph. And look, even without knowing anything about blood, you can see that things are different here. If you look, that is different from that, which is different from that, which is different from that. And so you can see that there are differences in here. If we look at blood, we probably already know a lot about blood. Everybody in here has seen blood. You probably all tasted blood, because the first thing you do when you get a cut on your finger or something, most people stick it in their mouth. Well, if you notice that blood has sort of a metallic taste, well, that's because there's a metal in it, and that metal is iron. That's why it tastes the way it does. You probably know it's sticky. You can't see through it. It also varies in color tremendously. It goes from a bright, bright, bright red to a dark red, depending on the level of oxygen. pH is something else that's really important about blood. pH is how acid or how basic we are. Well, blood pH is very tightly regulated. Look at it. It does go up and down just like everything else, but it goes up and down like this, just a tiny little bit. Not like this, just a little bit. Look, the range is only 7.35 to 7.45. In other words, the range is 0.1. And if you get outside of this range, if you go this way, you're said to be acidotic. And if you go the other way, you're said to be alkalotic. Either one of these conditions is very bad. If it gets very far out of it, you can go into a coma and things like that. And if it goes below 7, you're going to die. There is nothing you can do about it. You're going to be dead. You can't recover from it. And guess what? If it goes above 8, the same thing. You're dead. You cannot recover from that. So look, that's just one pH unit that has associated with life. Outside of that, you're dead. Also, blood is very hot. Look, blood is hotter than normal body temperature. So if you cut yourself badly and you bleed on your skin, you can actually feel the heat from the blood. It's hot. How much blood do we have? Well, it depends on how big you are. It depends on your body weight. It's about 8% of your body weight. So if you weigh 150 pounds, you multiply that times 0.08. That's how much blood you're going to have. On the average, males tend to be bigger than females. And so males tend to have more blood. And so for males, it's about 5 to 6 liters. For females, it's about 4 to 5 liters. So if we take the average of all that, we say that the average is about 5 liters of blood. That's how much we have on average. And then if you think about why we have blood, why do we even have it? It's mostly all about this one word right here, transport. Look at all the things we transport, even heat, but nutrients, waste products, gases, hormones, all kinds of things. It's all about transport. And then another function is protection. And we're going to talk about pathogens a lot in a couple of chapters away. But the blood is one of the things that defends us against pathogens. Blood also is responsible for maintaining and preventing blood loss. So it protects us from bleeding also. So those are the functions: transport, defense of our body against pathogen, and protection against bleeding. Well, when we look at blood, we see different parts. We see plasma. Plasma is the liquid portion of blood. And then we have what are called formed elements. Most of us would just call those cells. But it's not really correct, because even though we have different types in here, these are not really cells. They don't have a nucleus, so they're not cells. And these are just tiny little pieces of cells, so they're certainly not cells. So if you want to be correct, you don't say cells, you say formed elements. But there's three kinds of formed elements. There are erythrocytes, which most of us just call red blood cells. Look, there's that word, even though it's there, it's incorrect. There are leukocytes, which most of us just call white blood cells. And white blood cells are cells. And then there are thrombocytes, which most of us just call platelets. So when we look at it, we can see all those different things. So the liquid in here, all of this liquid in here, that's plasma. And then here are the red blood cells. Look how many of them there are. There are tons of them. Well, look at them. They look a little bit like donuts. And that's because that's where the nucleus used to be. It's not really a hole, though. It's a thin area. So the cell is shaped like this. And that thin area is what you see when you look here. And then these are white blood cells, or leukocytes. And then finally, these are platelets. So again, we have plasma, that's the liquid, and then we have these formed elements. Well, if you put blood, whole blood, into a tube and you centrifuge it, when you centrifuge something, the heaviest stuff goes to the bottom. And the heaviest stuff is the red blood cells. And so red blood cells are going to get packed down here, just packed like crazy. And then the next heaviest stuff is going to wind up next. That, it winds up in a layer called a buffy coat. And that's where the white blood cells are, and the platelets. And then the lightest stuff is going to wind up at the top, and that's plasma. So we get three fractions. So here are those three fractions. So the plasma, the buffy coat, and then the erythrocytes, or red blood cells. Well, look, if we say that this is 100%, we can get an estimate of what each of these fractions is by figuring out what percent it is of this total. Well, this person, look, the red blood cells right here is 45% of the total. That's called the red blood cell packed cell volume, or the hematocrit. And a hematocrit is actually an easy way to estimate how many red blood cells we have. And so it's sort of a quick and dirty way to see whether or not you have enough red blood cells, or too many red blood cells, or you have the right amount. This layer here is not very big. This is only about 1% of the total. So look, there aren't that many white blood cells, and there aren't that many platelets, not compared to red blood cells. And then, of course, this is the plasma, the liquid portion of blood. But look, it's not clear. It's yellow. It's sort of a straw-colored fluid. And that means it's not just water. There's a lot of other stuff in it. So if we look at it, here's a centrifuge tube. And there you can see the plasma, and there you can see that buffy coat, and there's the percent or the packed cell volume of the red blood cells. If we separate all that stuff out and look at it, here's the plasma, and here are those formed elements: red blood cells, white blood cells, platelets. And so we're going to talk about each one of these a little bit separately. So let's look at plasma. So plasma is the liquid portion of the blood, and it's the largest amount. Look, in this person, it's 55%. That's pretty normal. But when you look at what plasma is made of, it's mostly water. It's about 92% water. But that's got a lot of stuff in it. One of the things that's in it is there are a lot of proteins in here. Well, these proteins, almost all of them are made by the liver. And they come in different types. We have albumins. Albumins are big proteins. And they're about 60% of the total. We have globulins. One of those types of globulins are called immunoglobulins. An immunoglobulin is just another name for antibodies. So you heard a lot right now, especially because of the coronavirus and all that stuff, about antibodies. So they're proteins. They're floating around in the plasma. They're called globulin because this is their shape. They're sort of glob-shaped. So we have immunoglobulins. But there's other globulins too. There's globulins that attach to things. Remember we talked about hormones and we talked about steroids. They had to have a carrier. So these globulins are the steroid carriers and the T3, T4 carriers. And so you find those in there. And then one other protein that you find in here is called fibrinogen. Fibrinogen is the clotting protein. Now in this state, it's not clotting. It's not a clot. It has to be converted to something called fibrin, and that's what forms the clot. We also have a ton of different solutes in here. Look, there's over 100 different solutes. I'm not going to go through all these, but look at them. There are all kinds of things: glucose, amino acids, sodium, potassium, chloride, oxygen, carbon dioxide, urea, creatinine. There's a ton of different solutes. A lot of these are ions. And if you look at ions, ions have a charge. And we've spent a lot of time talking about, say, sodium and potassium. Remember, they're called ions because they're charged particles. And so you can see that they have sort of an electric ability or an electric nature. So they're called electrolytes. So you hear about electrolytes. In fact, you probably drink like sports drinks and things like that. Well, sports drinks have a ton of these electrolytes in them. Here are all these electrolytes. So that's plasma. Let's look at the formed elements. Remember, those are the erythrocytes or red blood cells, the leukocytes, the white blood cells, and the thrombocytes or the platelets. Well, just like I said before, technically you can't use the word cells because not all of them are cells. Red blood cells are not cells. They used to be, but they're not anymore because they have no nucleus and they have no organelles. They're basically just a little bag. That's all they are, is a little bag that's floating around. And because they have no nucleus or organelles, they can't do a lot of things that normal cells can do. They cannot divide, so they can't go through mitosis. There's no DNA in there. They can't repair themselves. There's nothing to make proteins. They're just stuck with whatever they are. There are no mitochondria in there. And since there are no mitochondria, they cannot do aerobic respiration. They can't do it. So the only way they can make ATP is anaerobic. So they're not normal at all. And then platelets are not cells at all. They're just little tiny pieces of cells. So technically we should use this term, although most of us probably don't. But when you look at them, here are the formed elements. So there are red blood cells, there are white blood cells, and there are platelets. So let's look at these red blood cells. Now technically they're called erythrocytes, and I use that word sometimes, but most of the time I just say red blood cells, and that's perfectly fine for you to do as well. Remember, they look like donuts, but it's not a hole. It's just an indented area. Look at them. They're actually very pretty cells. Aren't they neat looking? They're really cool. They have a very, very characteristic shape to them. And the shape is called a biconcave disc. In other words, it's a disc, but it's sunk in. It's concave on both sides. Now, it wasn't always like that. The reason it's like that is because it got rid of its nucleus and its organelles. It actually sort of spits them out. It ejects them. They have a neat ability. Red blood cells go through capillaries. We talked about capillaries. Remember, red blood cells have to go through capillaries one at a time because they're about the same diameter. That diameter is about 8 microns. But some capillaries in the body are only about 3 or 5 microns. 3 to 5. So look at this. How in the heck is this 8 micron cell going to get through a 3 to 5 micron tube? Well, it has to do this. It has to scrunch down. It looks like a sausage going through there. Well, the only reason they can do that is because they're very, very, very flexible. They can bend, they can stack up, they can do all kinds of things. And the reason they can do it is because they have a protein in their membrane called spectrin. But remember, red blood cells don't have a nucleus, they don't have any organelles, and so they cannot fix themselves. So after about 4 months, they degenerate. So they only, and I'm going to put this in quotation marks, live, because they're not really alive, about 120 days. And toward the end of that 120 days, they begin to get very, very fragile. They get stiff, so they're not able to bend and flex the way they're supposed to. Well, remember, we said that most capillaries are about 8 microns, but some are about 3 or 4 or 5 microns. So which capillary do you think this cell is actually more likely to break in? So if they have to flex like this, a lot of times they rupture. And most of the capillaries that are like this are found in the spleen. So the spleen is sometimes called the red blood cell graveyard. because that's where they degenerate and break down. Makes sense? Anybody have any questions about any of that? So, here's what that looks like. So see the biconcave disc shape right here? And remember, here they are going through blood vessels, the capillaries. Look, they're so small. I mean, the capillaries are so small, they have to go through one at a time. They tend to stack up. And these stacked red blood cells, there's a name for this. Remember, it's called rouleau. Well, look how many red blood cells there are. There's a ton of them. Remember, they're not really cells. They're basically just a little sac, a little bag. And what a sac or a bag is full of is a protein. So there's a protein in here. There's just tons of it in here. That protein is called hemoglobin. And there's a lot of hemoglobin in every red blood cell. It takes up almost all of the space, about 95% of the space. And there's a ton of these hemoglobin molecules in here. In one red blood cell, there's about 280 million molecules of hemoglobin. That's in one red blood cell. Well, hemoglobin is a really, really important molecule. It's a protein. It's got 4 polypeptide subunits, 2 pairs. And in the center of those, so there's one here, looks like that. There's one here, looks like that. There's one here, looks like that. One here that looks like that. And in the center of each one of those is what's called a heme. So when you look at it, it looks like this. So look, you can see 1, 2, 3, 4, 2 pairs. There's 2 alphas and 2 betas. In the center of each of these is this heme. So that's a heme. And then the protein is a globular protein, a glob. So look, that's where the word comes from, heme glob, so hemoglobin. And don't forget, there's about 280 million molecules of this in every single red blood cell. Hemoglobin has a very unique property. Oxygen binds to hemoglobin, and it binds right there. So you can bind one here, one here, one here, and one there. So hemoglobin can carry up to, doesn't have to be, but up to 4 oxygens. There's the iron right there. Remember we said that blood has an iron taste. That's where it is. It's in the center of the heme. And that center of the heme, that iron, is where the oxygen binds. Nowhere else, just there. Well, remember we can carry up to 4 oxygens per hemoglobin. And don't forget, there's 280 million molecules of this hemoglobin in every single red blood cell, in just one red blood cell. So if I put an oxygen here, an oxygen here, an oxygen here, an oxygen here on every single one of those hemoglobins, this is how much oxygen one red blood cell can carry. About 1.2 billion molecules of oxygen. That's pretty amazing. That's just one red blood cell. And we have a ton of red blood cells. Any questions about hemoglobin? So where does the oxygen bind when it binds to hemoglobin? It binds to the iron, exactly. Nowhere else. And that iron has to be in the right state in order for it to work. It has to be in the 2+ state. So if something comes along and changes that, hemoglobin won't work anymore. Not only will oxygen bind to hemoglobin, but so will carbon dioxide. So oxygen binds right here. And when it binds, we call this oxyhemoglobin. So oxyhemoglobin is nothing but hemoglobin with oxygen bound to it. That's what it is. When the oxygen comes off, and it will, we call it deoxyhemoglobin, or sometimes just reduced hemoglobin. But carbon dioxide will also bind to hemoglobin. But it doesn't bind here. It doesn't bind to this part. It binds out here. So remember, these are peptides. Peptides are made up of chains of amino acids. And remember, amino acids have R groups. So that's where it binds. Over here, that's where it binds. And so when we talk about that, it's called carb for carbon dioxide, amino because it binds to those amino acid residues, hemoglobin. So carbon dioxide binds out here. Any questions? Not only will oxygen bind to hemoglobin, and carbon dioxide will bind to hemoglobin, but carbon monoxide will also bind to hemoglobin. Carbon monoxide binds here, right where the oxygen does. And what it does is it binds so tightly it won't let go. And it prevents the oxygen from binding. So if you get a little bit of carbon dioxide in your blood, every one of these that it comes in contact with, it's going to stick to it like glue. You cannot really get it off. And that makes this whole hemoglobin useless because it won't bind to anything else. It won't bind oxygen. So even though it's floating around in your blood, it's useless. And if you have enough carbon dioxide, you can do that to a lot of the hemoglobins, and you can die from lack of oxygen or asphyxiation. Well, when carbon monoxide binds to hemoglobin, it's called carboxyhemoglobin. So these are some names that you need to learn. Oxyhemoglobin, deoxyhemoglobin, carbaminohemoglobin, and carboxyhemoglobin. Any questions about that? And then remember, we have a ton of red blood cells. So if you count how many red blood cells in 1 milliliter of blood, in males, it's more than it is in females. But look, it's the range is right here. And here's the range for females. Anybody have any idea why females might have less red blood cells than males? Any idea at all? It's not that they're smaller. I mean, that has to do with how much blood they have. Because remember, that's about 8% of their body weight. So it has nothing to do with body weight, though. It isn't the blood, it's the red blood cells. Females have fewer. Think about what happens with females that does not happen with males. What happens with females? Usually between the ages of about 12 and maybe 50. Yeah, menstruation. And think about what happens every single month. If you have menstruation, you lose blood. Well, if we just look at the average across all people, we just say it's about 5 million. So these numbers are pretty easy to remember. 5 liters of blood, 5 million blood cells per milliliter. This number 5 shows up quite a bit. Any questions? Well, look, we want to keep the number of red blood cells constant, but remember they wear out. Do you remember how long they last? It's about 4 months or about 120 days. But they don't all die on the same day. They die constantly, and we're replacing them constantly. On the average, we lose about 3 million red blood cells every single second. 3 million. So we're going to lose 3 million, and we have to make 3 million in order to keep our blood cells a constant. And so it's a constant process. Well, red blood cells are made in red bone marrow. That's also where the other cells are made, but red blood cells are made in red bone marrow. That process is called erythropoiesis. What's interesting about this is it needs to be regulated. We don't want too many red blood cells and we don't want too few. So we need to regulate it. Well, the organ that's in control is the kidney. The kidney. The kidney produces a hormone. That hormone is called erythropoietin. It's just abbreviated as EPO. So the kidneys release this EPO. It goes to the red bone marrow and increases the production of red blood cells. So how do the kidneys know when to do it? Remember, the cells are carrying oxygen and they're delivering oxygen to your tissues. And so what happens is, well, let's look at this part first. So here's the red bone marrow. And in the red bone marrow, we have stem cells. And the stem cells that form blood are called hemocytoblasts or hematopoietic stem cells. And look, depending on what hormone they get, they can become any of the red blood cells, white blood cells, platelets. They can become any of it. So you have to give it the right hormone. And the hormone that makes it turn into red blood cells is this EPO. Again, that comes from the kidneys. What it does when it gets there is, there's our hemocytoblast. It makes it go into a pathway to become a red blood cell. And that happens in stages. So the first stage is we build a bunch of ribosomes. So look at this. Here's our hemocytoblast. We're going to give it EPO. It's going to turn into, eventually, a red blood cell. It's going to go through stages. And the first stage is we're going to build a bunch of ribosomes. Ribosomes are like machines. They're basically machines where we assemble proteins. That's what they do. That's all they do. They assemble proteins. We take the messenger RNA as a blueprint, and we're going to use amino acids as our raw material, and we're going to assemble them together to produce proteins. So the first thing we need to do is build ribosomes, and that's the first stage. And then the second stage is we're going to use the ribosomes to build the protein, and the protein is hemoglobin. So we got all the ribosomes built. We're going to start building hemoglobin. And look, the color is going to change. Hemoglobin is red. So the cell is going to go from this color to this color to this color as the more and more hemoglobin is built. And then the last stage is we're going to get rid of all of the stuff inside cells except the hemoglobin. So we're going to eject the nucleus, eject the organelles. We're just going to spit all that stuff out. And then we're going to be this cell here. So 3 stages: ribosome synthesis, production of hemoglobin, ejection of the organelles. Make sense? Any questions? And remember, we're making about 3 million of these per second. So the red bone marrow is a busy, busy, busy place. This is turning into this, and this whole process takes about a week. So you have to start a week before you need it. And then we're going to make these red blood cells. Well, remember, we don't want too many red blood cells, and we won't want too few. Either way, we're going to wind up with different problems. So we have to control the number. And again, that's the kidneys that do it. So the kidneys are going to release this EPO, and that's what's going to cause red blood cells to be produced. Well, here's the way it works. It works on oxygen levels. So if we have enough red blood cells, we're going to have enough oxygen. It's going to be just like this. But if red blood cell number falls, oxygen level is going to fall. And when the oxygen level falls, the kidneys are going to detect it. So as the blood flows through the kidneys are going to detect this reduced oxygen level. And what the kidneys are going to do in response to this reduced oxygen is they're going to release this EPO. And then EPO is going to go to the red bone marrow, and it's going to stimulate the red bone marrow to produce more red blood cells. And so if we have more red blood cells, we're going to have more oxygen. And so oxygen is going to go back to a normal level. Does that make sense? Any questions? So, low oxygen, kidneys are going to detect it. They're going to release EPO. That EPO is going to go through the blood, gets to the red bone marrow. Red bone marrows are going to start producing red blood cells like crazy, and they're going to enter the circulation, and they're going to circulate. Remember, they last about 120 days. Any questions? That's where we're going to stop for today. So we'll pick back up here next time. Any questions?